Hi everyone, let's learn about bacterial culture media. How the bacteria are grown in the labs. Just like humans need food, bacteria also need nutrients for their growth and nourishment. The special food which is required for bacterial growth and survival is known as culture medium. This food contains water, nutrients, and essential elements. Later on, we will see that which nutrients are used in different medias and what essential elements are required. There are different types of culture media because different media are used to grow different types of bacteria. Let's see the different types of culture media. Culture media are classified into general purpose media, enriched media, selective media, differential media, transport media, anaerobic media, specialized media, indicator media, enrichment media, and reducing media. So there are different types of medias for different bacteria to grow in the lab. What are the general purpose medium are used for? General purpose media, it supports the growth of broad range of microorganisms without favoring or inhibiting any. So these are the media that are used for general purpose. It will allow the growth of many organisms, but it cannot favor or inhibit the growth of any bacteria. So this medium generally it contains the basic nutrients. These are peptones, amino acids, vitamins, and sometimes blood or serum. Let's see the examples of the medias that are used as general purpose media. The examples are nutrient agar, nutrient broth, tryptic soy agar, brain heart infusion agar, brain heart infusion broth, Lori Bertani agar, Lori Bertani broth, plate count agar, Mueller Hinter agar, and cooked meat medium. What is enriched media? These are the media that provides extra nutrients to support the growth of fastidious bacteria. What are fastidious bacteria? These are the bacteria that are difficult to grow in the lab because they require complex nutritional or environmental needs. Examples are Neisseria gonorrhea, Campylobacter species, Bordetella, and many more. So what it requires as a food? This medium contains basal medium, which contains carbon, nitrogen, salts, and water. And then this is the basic medium. And then they are further enriched because these bacteria require extra nutritional and extra elements. So it contains blood, some of them contain serum, and some of them may also contain the growth factors like vitamins. The examples of enriched media are blood agar, chocolate agar, Loeffler serum medium, brain heart infusion agar, brain heart infusion broth, Mueller Hinter agar, Thayer Martin agar, egg yolk agar, buffer charcoal yeast extract agar, cysteine tryptic agar. What is selective media? These are the media that will inhibit the growth of unwanted microbes, and it will allow the growth of specific microorganism which is specifically required to be isolated. So this medium will contain basal media, which contains the carbon, nitrogen, salts or water, and then it will contain the additional elements or nutrients that will specifically inhibit the growth of other microbes and will allow the growth of specific organism which is to be identified or isolated. So these nutrients are selective agents, that is sometimes in some media there will be bile salts, some requires dyes, in some antibiotics or high salt, distilled water and agar. If the solid medium is required, then agar is required. Let's see the examples of the media that are used as selective media. These are McConkey agar, mannitol salt agar, XLD agar, thiomartin agar, Hyoactin enteric agar, cetrimide agar, scurose medium, bismuth sulfate agar, Lowenstein-Jensen medium, tellurite blood agar. What is differential media? These are the media that will distinguish the microbes or bacteria based on visible changes, sometimes on the basis of color and sometimes on the basis of pH. So it will differentiate between bacteria or microorganisms on the basis of their visible color change and sometimes pH change. So this media contains basal media that has carbon, nitrogen, salts and water in it, and the differential agents are carbohydrates like lactose or mannitol, sometimes pH indicators like neutral red or phenol red, and distilled water and agar for the solid medium. Examples of the media that are used as differential media: clad agar, mannitol salt agar, XLD agar, eosin methylene blue agar, McConkey agar, blood agar, chromogenic media, bile esculin agar, triple sugar iron agar, and Hyoactin enteric agar. What is transport media? These are the media that maintain the viability of microbes or bacteria during transportation without promoting the growth of unwanted bacteria. It also prevents drying, suppresses contaminants, and also maintains the pH of the sample. So these media contains buffer solution that actually maintains the pH of the samples, salts that preserves osmotic balance, reducing agents that is thioglycolate for anaerobes, gelatin or agar that prevent drying stabilizes specimen, and sometimes the charcoal, and it's optional that it absorbs toxins. For example, it is already used in Ames medium. Examples of the media that are used as transport media are Ames media, Storrs medium, Caribbean medium, viral transport medium, thioglycolate broth, buffered glycerol saline, CPC medium, Dubus medium with antibiotics, universal transport medium, and liquid Storrs medium. What is anaerobic media? These media are used to support the growth of obligate anaerobes by eliminating oxygen and maintains the reducing environment for the microorganisms or bacteria. So what are the nutrients that are used in this media? These are nutrient base like peptones or yeast extract. Reducing agents in such type of medias are thioglycolate and sometimes cysteine. Buffers are used to maintain the pH. Agar is used for the solid medium, and racezorin are used. They're optional. Like in some media, they are used for redox indicator to show the oxygen presence. Examples of the media that are used as anaerobic media: thioglycolate broth, cooked meat medium, anaerobic blood agar, egg yolk agar, kanamycin, vancomycin lact blood agar, phenyl ethyl alcohol agar, Schedler anaerobic agar or broth, brain health infusion agar or broth, cycloserine, cefoxitin fructose agar, and bacterialized bile esculin agar. What is specialized media? These are the media that are used for specific diagnostic or research purposes. So they use the spatial nutrients or elements to isolate or identify the specific microbes or bacteria. What are these nutrients or elements that are used for the specific detection of certain microbes or bacteria? These basic nutrients: peptone, beef yeast extract, NaCl or water. Selective agents: in some media they are used as antibiotics or dyes. Differential agents that are used are pH indicators or carbohydrates. Enrichment substances are blood, serum, or vitamins. Other specific additives tailored to diagnostic or research needs. Like these specific additives, sometimes for research or diagnostic purposes, we need if we are working on a novel bacteria or identification of a novel microbes, then definitely according to their needs, the specific additives are added. The examples of the media that are used as specialized media: Lowenstein-Jensen media, also known as LG media; Middlebrook 7H10, 7H11 agar, buffer charcoal yeast extract, cysteine tellurite agar, chromogenic media, selenite F broth, tellurite blood agar, Campibap, thiosulfate citrate, biosars, sucrose, most commonly known as TCBS agar, and XVLD agar. What is indicator media? These are the media that detects the bacteria on the basis of biochemical activity through color change. As it uses the specific indicator which causes the color change depends on the type of the bacteria. So which nutrients or elements are used in this type of media? It uses basic nutrients, for example, peptone, beef extract, NaCl, water. Indicator agents are used. Which type of indicator agents are used in different types of indicator media? These are pH indicators like in some media phenol red or bromothymol blue or neutral red. Chromogenic substrates and ferric salts are used in some media. Differential substances that will differentiate on the basis of color. Carbohydrates like lactose and mannitol and amino acids. Other additives that depends on the specific type of bacteria. These are tailored for detecting specific bacterial enzymes or reactions. So sometimes the other additives are also added that are designed for the detection of specific bacterial enzymes. Examples of the media that are used as indicator media: McConkey agar, mannitol salt agar, XLD agar, Simon citrate agar, hectoin enteric agar, urea agar, bile esquilin agar, chromogenic media, triple sugar iron agar, and eosine methylene blue agar. What is enrichment media? These are the media that will allow the growth of specific bacteria that are often present in less numbers in the sample that are arrived in the laboratory and will suppress the growth of other unwanted bacteria. So this media, it contains basal nutrients, sometimes peptone, yeast extract, NaCl. Selective agents are used chemicals that will inhibit the unwanted bacteria. These are biosols, selenite, and tetrathionate. pH adjusters to favor the growth of target organisms, that is alkaline pH for Vibrio. Water for solvent and for providing the environment for the growth. Examples of the media that are used as enrichment media: selenite F broth, tetrathionate broth, alkaline peptone water, gram-negative broth, mannitol selenite broth, buffered peptone water, Fraser broth, Rapoport vacillidis broth, Todd Hewitt broth, and GN broth. What is reducing media? These media are also used for the growth of anaerobic bacteria by chemically removing oxygen with the help of reducing agents. So these media contains basal nutrients in the form of peptone or yeast extract and NaCl. The reducing agents are sodium thioglycolate and cysteine. Buffer system, which maintains the pH. Risazorin, as it's optional, it is used for Radix indicator. And agar to provide the semi-solid medium, as it limits the atmospheric oxygen to be diffused into the medium. Examples of the reducing media include thioglycolate broth, cooked meat medium, brewer anaerobic agar, hectoine enteric agar, anaerobic blood agar, enriched thioglycolate medium, peptone yeast extract broth, neomycin blood agar, cycloserine, cefoxitin fructose agar, and chopped meat glucose broth. This is all about what is culture media, the classification, the major classification of culture media, and then the different types of culture media. There are further series of videos in which each culture medium that is mentioned in this video are explained in details, in which for each culture medium, the purpose, composition, preparation, and the type of growth on the media, all these will be explained in separate detailed videos. Don't forget to watch them, and thank you so much for watching.